assuming that we were all able to edit all of those files to generate 4 new sequence count files, what we would have is 4 sequence count files, all of which look pretty much exactly like the one that I showed you earlier, which is gene name count. What do you do with that? Right? What are we trying to do with all of this data? What we're trying to do is get a list of differentially expressed genes. So on a superficial level, you could see very easily how what you'd want to do is take every gene, look at the average of the number of counts for that gene in one condition, compare it to the average number of counts in the same condition, or of the same gene in the other condition, and find ones that fall above the threshold of fold differential change or statistical significance or what else you want to do. Here's the problem. In most cases, you're going to see something sort of like this if you were to average out your counts. If we assume that the genes in this sample are A, B, or in this organism are A, B, and C, and we have condition 1 and condition 2, what can you actually say about these 2 samples? Can I say that gene A in condition 1 is significantly upregulated over condition 2? So what are these numbers? These are absolute gene counts. That means of the number of reads that we aligned to the reference from each one of these samples, that's how many aligned to that gene. So by the same token, could I say that sample or gene B is significantly upregulated over gene or condition 2 over condition 1? Not right now. And the reason is this. How many reads are in my sample over here? About 120, 100,000, a little bit more, versus a million. I have a million more reads for all intents and purposes, 900,000 more reads in this sample than I do over here. So before I can make any comparisons between condition 1 and condition 2, I've got to have something close to the same number of reads in both conditions. Right? How do you do that? Well, you have to normalize. So before you normalize your data, any comparisons between samples is null and void. The only thing that I can say about these samples before I start applying normalization protocols and other things is that gene B is significantly upregulated compared to gene A in both samples, or both conditions. Because I can't compare between conditions until I have some sort of normalization factor between them. Now, for the vast majority of experiments and the vast majority of people, you were told to target 50 million reads per sample. So you're going to be kind of close. However, we saw in the first 2 samples that we were looking at that you had a difference of about 10 or 15% of the reads that survived QA and QC between lung and saliva. So that means that it's likely that you're going to have similar problems when you get to comparing the gene expressions to each other. The take-home of that is don't try to do it from raw numbers. And really, given the amount of sophisticated statistical analysis that is available in the world, don't try to do it yourself is probably the secondary message of that. Let one of the tools that exists do it for you. If you're using EdgeR, let EdgeR do the work and then figure out what the answers are. Don't try to figure out exactly how it did the heavy lifting, unless you really care and you want to get a second PhD in statistics. The short list of things that I know of offhand that are going to be important for calculations of differential gene expressions. The relative counts of genes in the sample, that's the one that we were just talking about there. Secondarily, standard deviation of read counts within a condition, both really, within a single condition. So if you have 3 replicates and gene A has a significantly larger standard deviation or coefficient of variation than gene B, it's going to be harder to determine when something is significantly up or down regulated compared to the other condition. I just said a lot of words. Do they make sense? I'm not seeing heads showing up and down very often. I'm going to say it again slightly more simply, I hope. if you're comparing between a single gene and in condition 1, that gene has a very wide spread of expression values. Sample 1 is 500 counts, sample 2 is 5,000 counts, sample 3 is 50,000 counts. The average of that gene expression is going to be about 40,000, right, or 25,000, whatever that number is supposed to be. However, the standard deviation, the coefficient of variation of that gene is very, very wide. It makes it really difficult to compare to another gene unless it is really, really differentially expressed compared to this, right? If it's expressed at 1 million counts per gene, then it's going to be pretty easy to make that call, but that's because of the fold differential, not because of the standard deviation, right? So they call it standard deviation, a coefficient of variation. When an individual gene has a lot of variation, it's harder to make a call as to whether there's differential expression or whether that's just noise. By the same token, if a sample, or if sample to sample, a lot of genes have a lot of variation, it's harder to be sure whether that variation is individual to a sample or is difficult to call for the whole condition, or what. So all of these call into difficulty in making a differential gene call. The total number of reads per sample. The more reads you have, we talked about this earlier, the easier it is to say for sure that a sample or a gene is really expressed or is really expressed at a given level. You get more confidence the more reads you have. The number of samples per condition is also going to kind of fit into that set. So to account for all of that statistics being different and the fact that everybody who uses p-value has been doing it wrong since the beginning of time, EdgeR and most gene differential callers don't use p-values. Let me rephrase that. They calculate the p-value and then they tell you to ignore it. So I'm going to do the same thing. I'm going to tell you to ignore the p-value. P-value is useless, especially in differential gene expression. There is a wide body of literature on this. My favorite one is a review paper from 3 or 4 years ago saying basically all of science is wrong because of p.05. If you're analyzing the human genome and it has about 500,000 genes, or 25,000 genes in it, and you use a p-value of 0.05, fundamentally that means that you have gotten at least 5,000 or 500 genes wrong. Just by virtue of the p-value being 0.05, the corollary to that is that 5% of your calls are wrong, including your negatives. It's not really a great number when you do lots of high throughput stuff. The statistic that they use, which is related to the p-value but is for gene differentials, is called the FDR. It is not short for Franklin Delano Roosevelt. It is, in fact, the false discovery rate. There's a pretty good Wikipedia page on it. If you want to follow the link later, you can. It's giving you the chance, regardless of the number of reads or the number of genes that you're looking at, that the particular call that is being made there is a false discovery. If a gene is differentially expressed according to your statistics, it will tell you that that false discovery rate is a number that is not quite directly correlated to the p-value. So I recommend highly when you get charts out of EdgeR or DE-Seq, or I think CuffDiff gives it as well, you use the false discovery rate rather than the p-value. You'll have to explain it every time you give a presentation to people who aren't used to it, but it's worth it. You can give them the p-values in the presentation if you want to, although I'd recommend against that as well, because then they'll ask you questions about why you picked one with a p-value that was so high when the false discovery was so low, and then you have to explain it all over again. All of that being said, and all of the work that I had you do this morning, we are going to pretend you did again, and we're going to copy and paste all of this to grab all of that data pre-calculated from somewhere else and put it into a new directory and do some of the work. You'll see something that looks a lot like this. It's going to go through and download a bunch of things. And what it is doing now, curl is a command to download information from the internet. And so all of this has been deposited in the old read the docs page that Titus built to go grab all of the data and an R-script file. And the last thing we're going to do is actually apply the R-script file, which is going to print a bunch of things out onto the screen. So I mentioned that this is using EdgeR. EdgeR is a tool written in the R statistical programming language, which again adds a layer of new things that you get to learn to deal with all of this stuff. So you've already learned a little bit of bash, a whole lot about RNA-seq, 4 or 5 of the tools required for RNA-seq, and now there's EdgeR. EdgeR is written to be, or is a programming language essentially. It has a bunch of functions that you can apply within EdgeR to do different analyses. There are a couple of things, and we'll look at the actual code in a minute. First, it prints out genes just so that you can see what it looks like. And then it prints out the table of all of the genes, or the first 5. You'll see here it's the same 6, excuse me. But now it's looking at them between all 4 conditions. So it took all of your data. You can see it took one condition of your data here, your lung replicate 1. And I made a table where it has every gene and then every condition next to it, or every condition as a column in that gene. So it's essentially making a table. Does that make sense? Right? You can't do comparisons between things without putting them into a table, at least in stats you can't. And then it did a bunch of math. It loaded some packages and then told you some things. All of this is output that is meaningless to you. It just means that it finished. But what it did do is make a new set of files, actually 2 new sets of files. So the things that we downloaded here are all of the replcounts.txt files and the lung saliva R file. So anything else is new. So these 2 files were made by EdgeR. So let's take a look at the PDF file. By the way, tab is a wonderful tool in bash. If you start to type and you know that you're looking for a file, so I know that I'm looking for a file that starts with ed and I hit tab, it completes as far as it can, and then I say, well, then it was MA and I hit tab again and it all shows up. Tab is a wonderful tool. So in a second, GB is a PDF viewer, and you can see it showing up over here. What does it show you? Here we have a really pretty chart. So if you were going back to basic principles of do something to get other people off of your back, the first place where you can do that when you get sample data, somebody will say, well, you have sample data, what have you done? You can make your FastQC file report and hand it to them and say, I did this. And then they'll leave you alone for about a week, and then they'll come back and say, well, now that you have all your data trimmed and your QC done, what else have you done? And you can show them this chart. And say, this is a chart of my differential gene expression. Which it is. The format of this chart is twofold. One is the log fold change compared between 2 samples, and the x-axis is the number of counts per million, which is a weird calculation that somebody figured out a long time ago. It's CPM, not log CPM, what's what I'm looking for? RPKM value, if any of you have heard that. Reads per kilobase per million reads, per kilobase of reference per million reads. It's the equivalent here. So what we're looking at is a chart that is, for all useful purposes, showing us on the x and y axis whether a gene is up or down regulated on the y axis, and on the x-axis how highly regulated that gene is at all. So what the average number, or what the average number of counts for that gene is overall. So The farther this way you go, the more reads there are associated with any given gene in this pool. The red and black coloring is a filter that was put in place to find genes that EdgeR believes are significantly differentially regulated given a cutoff of the FDR. So we're looking for a false discovery rate below, say, 0.05. What do we find? I have an interesting point here. If you've got it open, it may be easier to see on your screen. But where the cursor is right now is sitting on top of a little red dot. No, it's next to a little red dot. Right there. That little red dot doesn't make sense to the naked eye because it is very, 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 very exceptionally close to a zero differential fold expression change. But EdgeR says, I believe this to be significant. Why is that? Not only do you have a lot of reads, because there are a lot of reads, there's the covering over the T here. There are more reads here, and it's a log scale, so this is a lot more reads here. It's closer to zero, but it's still a lot more reads. There aren't any good ones here. There will be times, too, where you'll see one out here that's an outlier that's not red, right? That the FDR fails. So what do we think? How can you get those weird bits and pieces like that? Thinking back to my list of reasons how it calculates FDR. Coefficient of variation is the short version of that same answer. The smaller the variation, the easier it is to make a call. If all, in this case both, of the samples have a very close call, and all of the other conditions have a very close call, even if they're only 15% different, if it falls outside of the error bars, or the expected error bars, it's something that you should consider a true differential. I want to point this out, and I want to take the time to talk about this because there are a lot of people who have learned how to do biology from microarrays, and from before microarrays when all we had were qPCRs and QRT PCRs, and the only way we could be sure that we really had good statistics was if we had a minimum of X-fold standard variation, or variation between my two condition types. These are the people who will never, ever, ever want you to use that, and they're wrong. Right, and I fall into this category sometimes. It's not a large variation, so I shouldn't care about it, except the size of the variation doesn't matter. It's the significance of the variation is what matters. If it's real, and the statistics suggest it's real, you're not going to publish this paper no matter what until you go back and prove it. If you're going to have to go back and prove it, and this gene is showing up, and it looks like it's important, and it actually falls into a pathway that you think might be of value to what you're analyzing, don't throw it away because the variation is low, or the fold change is low. That's terrible science. That's throwing away data because you don't think that the results that you got from statistics are accurate. If you're going to use statistics, believe the statistics. If you're not, that's fine, but bear in mind that to a degree you're waving your hands and telling other people to believe that you have not just hidden a rabbit in your hat. So the other thing to look at is the actual outputs. So that's what you show the person who is on your back asking you why you haven't done any work in the last week. And you say, well, I've been looking at this chart and trying to figure out what to do with it. You can try to figure it out if you want. And then while they take a look at that, you can actually look at the table, which is important. So EdgeR outputs a table that gives you your gene expression. It also, as a really helpful additional thing, sorts in reverse order by the FDR. So the lowest FDR genes come first so that you can actually quickly filter and look for the things that you're interested in. Beyond the fact that there were what looked to be hundreds of different genes that showed up as differentially expressed in your samples, you can see that there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 genes with an FDR of 0, which is pretty good, indicating that at least given the reads that you gave it is 100% certain that these are different from each other. So the log fold change is in all of these is pretty high, except for the last one. And the log CPM is also really, really high. And the p-value is 0 as well as the FDR. This is great. This is really, really interesting. What do we do now? If these samples were real and if the number of reads were enough that I was actually willing to move forward with this analysis, what do I want to do next? Well, yeah, the pathways are a great figure. But to get to pathways, you need to know what the genes are. What are these genes? I can't tell you what the ensemble gene 17201 is. So I can't give you any results. But the next thing to do is probably to take your list of genes that you believe and see what you can find out about them. So there are a bunch of different ways to do this. The easiest by far is to use other people's web pages. Panther DB is easy. I won't say that that's the only reason to use things, but it's not a bad reason. Panther DB is a mechanism to look at gene ontology and pathway analysis if you give it, say, a list of gene IDs and an organism that you're working in. If I'm interested in these, this is the second time I have Excel opened today, which is more than usual for me. My brain has frozen. There we go. One of the adages that I was really always fond of, and I talk about or I use a lot as a statement, is that bioinformatics as a field came into existence when data became too large for an Excel spreadsheet. Biologists are really good at Excel. They tend to not be quite so good at command line because there's not enough time, or databases, or all of the other ways that you could handle data. So as the other side of my life when I do bioinformatics consulting work, the general question, if I were to break down almost every request I get for work, is, can you please take this and turn it into an Excel spreadsheet that I can use? So the end result of almost any analysis that you're going to do is going to be a spreadsheet with data that you can use. So once you've done all of this work on the HPC or on a Linux machine or something, sooner or later you're going to get some sort of CSV file that looks like this that you can import into Excel and actually do things with the data. So for the sake of speed, I just copied the first 10 of those gene names. You could actually download that CSV file, import the whole thing, and we might do that in a couple of minutes, but we're just going to look at the first 10 genes. So I'm going to take those 10 genes, and I'm going to paste them into Panther DB, tell it that it's an ID list, tell it that it's Homo sapiens, and then Panther gives me a couple of options as to what kind of thing I want to do. Given what I have, I really want, let's start with the gene list. I want the gene list of those identifications to see what I have. Given that this experiment was looking at the difference between salivary glands and lung tissue in some sort of condition, some sort of disease, what genes do we see that are differentially expressed? Well, we see mucin 7. I wonder what mucin 7 does. Anybody want to take a guess? That would be a pretty good guess. I'm not positive, but we're going to find out. Subfamily information, it's not in a family, but it's called mucin, so we can guess. We have a lysozyme, which is kind of general. We've got pulmonary surfactants. Let's see, pulmonary, lung, so far we're not doing too terribly, right? We're getting genes in our top 10 that are all things that I could really believe are differentially expressed between two conditions where one is the lung and the other is not the lung, but also happens to have a lot of mucus in it. So that gives you a start. And you can say, well, I've gotten the annotations of these genes. You can do a lot more with Panther DB. You can, as a bare minimum, look at charts. You can resort it based on what kind of gene ontology it fits into. We've got a gene that is a pulmonary surfactant that is also involved in immune system, another that's immune system, and a bunch of other. Here we can look at what genes are in that same pathway. Right, all of the genes that are involved in that pathway. And this is where you could, if you wanted to, start to build an idea of what story you're looking for. But at a bare minimum, that would give you the ability to let's go all the way back, sorry. Back to where we came from. I can just take this and export it to text and have my list of gene names so that I can take that as a table and hand it to somebody else and say, these are the genes that are differentially expressed. Pretty easy, right? Yep. So the next thing, so this was just, I just wanted to see what the classification of the top 10 were, and I was happy that those came out. But the more interesting thing in this, to me anyway, is if we are going to look at the whole sample. So I'm going to do this a slightly slower way. So if I wanted to, the next thing to analyze would be, assuming that I have a pathway, as I do with a model organism, I can actually grab the whole set of data and filter them, only selecting down to a certain FDR rate, not the top 10, but say we'll select everything below an FDR of 0.05. And we'll paste it into the same program, except we'll paste all of them, and then we can look at the next option, which is the statistical overrepresentation test. So what, while I'm waiting for my email to come in, would we care about, or would we think the statistical overrepresentation test is actually going to look for it? What do we care about when we're doing FDR? We want to see what genes are up and down regulated in a condition. Why are we trying to do that? What do we really care about? Because we don't care about individual genes usually. Yes, pathways. So how do you determine if pathways are up or down regulated, right? In the real world, biology is messy. A whole pathway is not going to be up or down regulated, right? The citric acid cycle doesn't have all of the genes turned up all of the time because they have enough of one of the proteins, but not enough of the other, and it keeps cycling through. So you have to hit that. So how you have to incorporate it more closely. So how do you find out what gene pathways might be more affected by a condition than others? The way that they go about it in this is to, so I'm going to take all of these and we're going to see what it gives us, and then we can figure out why it's doing it. So let's go down to an FDR of 10 to the minus 5, since that gives us 200 genes or so. Okay, paste. We're going to do statistical overrepresentation. It's thinking. So what is it going to do? It's going to take those 200 genes, and what is it going to try to figure out? It's going to try to figure out if you annotate those genes and put them into their respective pathways, how many of those genes fall into pathways versus how many genes exist in those pathways. And then it's going to tell you whether or not it believes that there's some degree of overrepresentation of those pathways. Unfortunately, not everything's useful. So Panther gene ontology, slim biological processes, or which processes they are. Antigen processing and peptide processing. It says might be overregulated. And the reason it thinks that is that the reference has a total of 38 genes that are involved in that process. The input out of the genes that I pasted in, the 200 or so genes that I popped into this list, 6 of them were in that category. So it's about 5-fold enriched versus the number of genes versus the total number of things I pasted in. Is this starting to make sense? I'm going to go over it again just to make sure. So the next one down, well, of course, my favorite of all of these is unclassified, right? So just to show you that not every process, just because you get significance, is useful, we have a p-value of 0 for unclassified processes. I am absolutely certain, says Panther, that unspecified processes are over-enriched in your differential gene expression. We can all go home. We have found the solution to our problem. We need to fix the unclassified gene sets in the human genome. Given that that's not the case, right, you can see really that respiratory electron transport chain may or may not be useful. Oxidative phosphorylation, same way. Regulate. So here's an interesting one, right? Liquid surface tension and antigen work, given that we're dealing with disease conditions and the lungs. Both of those might be very, very interesting, and 5 genes gives us a greater than 5-fold upregulation and a pretty decent p-value. It's 0.01. Right? Yeah, 0.01. So all of these are ways that you can mine that database. So you can also, if you really care, once you've pasted all that in, click on the link and give me a list. And you can see more information about it. It'll tell you all of the families, subfamilies, et cetera, et cetera, et cetera. And you can make all sorts of pretty pictures. Which, again, is not a terrible thing, right? Pictures are great. The only statement I will make after that is that I am highly in favor of finding ways to get a website or a tool to make charts for you, because you need charts for publications, because it is way easier to describe information in a chart than it is in almost any other format. They are completely useless for analysis. They are the end result of analysis. They almost never give me, at any rate, any useful information while I have them. I'm making them because I already know something and I need to describe it. So charts should always be your end goal, but are based on understanding and information that are going to come from all of the other stuff that you have to do first. So bear that in mind as we go through this. Okay, so let's take a little bit here and look at EdgeR. After all, we want to know how we actually got this data out. So we're going to look at the nano of lung saliva .repl or .r to show what we're looking at. So if you see files that end in .r, they're always going to be using the R statistical programming language. That doesn't always mean that they're using EdgeR. There's an equally as useful but slightly different set of statistical tools called DE-seq that does differential gene expression analysis, differential expression out of sequencing, which was built, I think, de novo for sequencing only as opposed to EdgeR, which, as I probably mentioned, because I know in my head I'm not sure I said it, was based first around microarray tools, was adapted to RNA-seq, etc. So there are two tools. There are people who are very, very strongly in favor of one or the other. There's no reason not to. So if you're doing RNA-seq and you see an R file or .R file, it's probably doing analysis in one of those two packages. So what are we doing? R is possibly the most confusing language I've ever tried to learn. It does not do things the way that most programming languages do them, which means that you have to learn it all by itself. It doesn't look like anything else. There are good and valid reasons for that. I'm not trying to say that it's a bad language. It's just written very differently, and it's written essentially by statisticians the way that statisticians think about things, and so they're structured slightly differently. Which is to say that it creates objects and then reads things into that object. So the first object is the name of the samples, and the little less than and a dash, which looks like an arrow now, is reading in a combination of the names of the 4 samples. So the first thing it does is it creates a variable with the names of the samples in it. If you had 6 samples, if you were going to use this same methodology, you would make a combination line with all 6 sample names in the order that you're going to read them in later, one at a time. The next thing you're going to do is try to make this really fancy function here, which is when you're going to read the sample, you're going to read into it the function of the sample name, which links to the file name, and all of these things, which basically is taking the name of these samples one at a time and appending counts.txt to them, and then doing a bunch of other stuff. So basically all of that function is doing is reading in each file. There are other ways to do this. I will happily show you tomorrow other ways to do this if you really care. I'm not trying to teach you EdgeR in detail right now because you've already had to learn 12 different things. So all of that means that when you want to do something to it, so now these 2 bits are required to be able to do this. What we're doing is we're making a function that reads the sample in, a list of sample names. So now we're going to make samples 1, 2, 3, and 4 by reading the samples 1, 2, 3, and 4. Kind of makes sense? Yeah? So all we've done really is we've made these new variables because of all this that are named sample.1, 2, 3, and 4 that read in the files for REPL1, REPL2 for lung and salivary. That's what we have here. Well, that's great. And then if you remember when we were looking at this, the first thing it did was it just printed the first 6 lines of sample 1 because that's what this is told to do. That's a function that heads prints the first couple of lines of a variable. And so I was just checking to make sure that we read it in right. What we have to do to actually do analysis is we have to combine the data into a single data set. We have to make a table with columns for each one of our conditions. And so all it's doing is it's taking the data and putting it into what they call a data frame, sample 1, 2, 3, and 4, with extra stuff at the end of that because R. And then, of course, we want to name them. So how do we name them? We say, well, we know that we read them in order. We read them in 1, 2, 3, and 4. And the names that we want those to be are the first function or the first variable that we created. So we're going to name them that, which is why we're reading samples into the column names of all the data. So now, when I head that table, that's how you get the list of the actual conditions at the top of the table because we did all of this work. Okay. All of this is data preparation, getting your data into a format that EdgeR can use. This part, there are lots of different ways to create a data structure that works. Like I said, I will happily show you one that I've written that I like a little bit better tomorrow. But this way at least sort of gives you an idea of how it works. Once you have all of the data, you have to actually start doing EdgeR. So EdgeR is a library. It loads it, and then it has to group them. So what do we know about all these samples? Well, we know that there are 2 conditions. There's lung and they're salivary, and they have 2 replicates each. So we're going to group them by replicate type lung or salivary, which we created because they have the name lung or salivary in their names. And then in here, it separated everything out so that it would show you which one is which. EdgeR has a bunch of functions, and by the way, of documentation of bioinformatics tools, I'm going to take a slight step sideways because I have a chance to stand on my soapbox here. Documentation is really the most important thing you can do if you're writing tools. Bioinformaticians in particular are terrible at it. They will write tools and either write far too much documentation in the output of the command line when you write it wrong, or put nothing anywhere except run this and it'll work. It's awful. One of the few exceptions to all of that whining and complaining that I will do about it is the EdgeR user manual. It is 100 and something pages long, written in a way that you can actually scroll to the kind of analysis that you want to do and copy and paste the recipes, and it will work more or less out of the box, and then explain why it worked out of the box. It's shockingly useful and very, very nice. So if you're going to write new R code to do things, use the documentation in their manual. It's great. It has recipes, it has examples of different kinds of things. It works out well. All of that to say, there are really 2 different kinds of analysis that you can do using EdgeR. There are very few reasons not to do the DGE style, which is short for, unsurprisingly, differential gene expression. Differential gene expression expects 2 conditions. If you're doing experiments that have more than 2 conditions, you will want to use the other one, which I cannot remember off the top of my head, but I could go dig it out. That is not differential gene, it's not DGE, it uses a different set of functions to perform the same tasks. But differential gene expression assumes that there's a group and that you have a table of counts, which is what we have. And so the first thing we want to do is look at the dispersions. Dispersion is another sort of way to talk about coefficient of variation and other things. Basically, the first thing you have to do anytime you're doing statistics on things is to figure out how much the actual sample varies within itself and how much variation there is between samples, which is a very handy function, or set of functions right there that you tell it, estimate the dispersion, and I'll be done. Once you get all of that, the only other things you have to do is do the analysis, and then graph it, and then output it. That's really all it's doing here, is it's doing the exact test, which is saying, I have 2 samples, or 2 conditions, do an exact test to tell me whether they're different from each other, and then it's grabbing the top, in this case, 100,000 genes. If you have more than 100,000 genes that are differentially expressed, this is going to miss some of them. If you have over 100,000 genes that are differentially expressed according to your conditions, I pity you. And the fact that you're going to have to go into EdgeR and change that number to be able to get the full list is the least of my concerns for you. It means that every gene in your sample or in your organism were differentially expressed between your 2 conditions. Again, it outputs all of that sorted by the FDR rate. And then it simply takes those 2 pieces of information and makes a PDF plot here, which at a FDR, if I can use the right arrow key, less than or equal to 0.1. So everything that was colored red in that chart was at an FDR of 0.1, which I would not recommend for real analysis, but because we used a small subset of the samples, it was the best. If we use 0.05 or 0.01 where we should be, you get 2 red dots and everything else is black because there's just not enough data there to analyze. And then it says if it's over that number, then it color it red. If it's under that number, under that number, color it red. If it's over that number, color it black. And then prints all of that to a field. It prints out this way on the table because there's a typo in there that I haven't bothered to fix, so that it actually doesn't print with the $ signs and makes more useful labels. But these are just the labels on the chart here. And then the last thing it does is print out the table named whatever you want it to do. So this is actually all you have to do with EdgeR. You'll notice that really EdgeR in all of this is 1, 2, 3, 4, 5, 6 commands out of the 30 or 40 lines of code. So it's less than a quarter of all of the things that are in this, and everything else is involved in just getting R to take your data that you have generated somewhere else and fit it into R to be able to use EdgeR. Okay, so what is this EdgeR never taking into consideration? What other confounding factors can you have in RNA-seq that we're not thinking about yet? What we don't know about these samples, because of the way that I grabbed this, is are these samples actually independent of each other? How do you know that lung replicate 1 and saliva replicate 1 are from the... Let me rephrase that. Assuming that lung replicate 1 and saliva replicate 1 are from the same individual, and replicate 2 are both from the same individual, what potential problems are there with that statement? So when you're doing an experiment like this, I want to know what the difference is between lung and saliva. I have to sample from the same individual, right? Because A, there's a limited number of individuals I can sample from if I'm interested in people with a condition. But B, I'm also interested in if I just sample randomly from people and only take one saliva sample from a person and one lung sample from a person and they're different, what do I find out? There's much less power in that, right? If, on the other hand, you do take from the same individual, there's a secondary concern of a batch effect. So what does a batch effect look like in something like EdgeR? Or when you're in a more general case, what does a batch effect look like for differential gene expression? What would that mean? This is not in the EdgeR code that I wrote, but this is the way that you can actually check for your batch effects. So it's a tool called plot NDS. Fold change in dimension 1 looks a lot, for those of you that have ever looked at it, a lot like a PCA plot, right? You're looking at two of the many dimensions of your analysis and trying to see something. So what are we interested in here? If you have a mock condition and an HRCC condition here, is there or is there not a batch effect based on this plot? What would it look like? Well, what you would think would be something closer to, I'm trying to find the actual plot of a batch effect here. Give me a second. So if we're looking for batch effects, right, what we would think we would see is that the samples taken from the same individual would be more closely related to each other than they would to other samples of the same condition. So if I'm sampling from lung and saliva, salivary glands of an individual and comparing it across the board, what I want is to see what genes are differentially expressed in the saliva versus what are differentially expressed in the lung, right? But if the differences between the individuals link much more closely than the sample type, I'm never going to see that. It's going to give me huge amounts of dispersion. Does that make sense? Is everybody with me? So how do you deal with a case where you have a batch effect given that? EdgeR has a way to deal with it, which is to say, do the analysis assuming batch effects. You don't want to do it if you don't have a batch effect, but you certainly don't want to ignore the possibility that there is. So what you have to do then is, first, you have to determine whether or not from a plot, because there isn't really any other good way to do it, whether or not there is an overall batch effect between the samples, and then if there is, you say, assume that there is, and then I want to see a differential gene expression by sample, so only look at the differential by sample and then combine those differentials together. So instead of averaging condition, first and then comparing. What it does is it compares with an individual first and then averages those differentials, right? So it just takes the analysis and flips it on its head. You don't want to do it when they actually group together properly, because then you're going to actually lose all of your sampling power. Does that sort of make sense? It's one of those that you only have to use it when you have to use it, but if you have to, you need to absolutely apply to have it. And the problem here, and the last thing that I really wanted to bring up in all of this, is that all of the things that I'm giving you in EdgeR are things that we are scripted here because we know it's going to work, but is almost always something that you're going to go through in iterations. This is the stage where you actually are going to run something, look at it, check something out, run it again after you change a couple of things. Those of you that use HPC regularly, or might be using HPC regularly, it's very easy to say, I'm going to run all of my analysis as Q-sub. So I have 55 samples. I'm going to submit them all to run. But then you need to wait for all of them to finish. And while you could submit your EdgeR stuff to run on the cluster, it's actually a really fast process, right? It took 2 minutes or 3 minutes to run on 4 samples. The amount of time it will take EdgeR to run is almost only related to the number of genes in your sample and the number of actual samples that you have. So it's always going to be faster to run than anything else. So really, you could script it to run everything together as another submission job and walk away, but you're going to be better off running it as a single test at the very end of your run and doing it all manually. You can't actually do everything that's scripted here. You can do interactively in R, sort of similarly to what you may have done if you were playing with Python and other workshops, where they are Python notebooks and those kind of things. Type one thing, hit return, it'll give you reply, and run through things individually, and then if you really want to run the analysis again, you can copy and paste through. So.